over eons of time, we as human beings hooked up not just with one another as cavemen, or way before we even became cavemen, but we hooked up with the organisms in our environment, what we used to call the commensals, or the normal species that lived with us. We hooked up with them and we internalized them. They became part of us, especially the gastrointestinal microbiome, which is the largest in the body, but we obviously have microbiome everywhere. So we developed this association, this relationship with the bugs in our environment, and we now call it a microbiome. It's become critically important to us as human beings in terms of outsourcing our immunity. We as a human species have given a lot of the control of our immune regulation over to the microbiome within us and on us. So let's consider what our microbiome is. This is the entire microbial content of an organ or organ system. The bugs themselves, remember, we're talking about more than just bacteria. There's a virome, there's a fungal microbiome, and there's a parasite microbiome in most of our organ systems. But it's clearly their genetic material that determines the anti-inflammatory or host response. The surrounding environmental conditions, the food and substrate, are important, and of course the interaction of these bugs with the human host is what's important. So consider this cartoon as an example of a healthy microbiome epithelium interaction. You can see that the epithelium is neat and intact, that the surface is covered by normal flora or normal microbial organisms. They interact with receptors on the epithelial cell, in this case toll-like receptor 2, which is the toll-like receptor that much of the microbiome uses to induce anti-inflammatory cytokine and cellular responses. And that's the important activity of the microbiome, to be able to create an environment not only on the epithelial surface, but for the systemic anti-inflammatory function of the human host. The microbiome is important to generate immunity for human beings. The gastrointestinal microbiome is clearly the largest in the body. It's the one that's easiest to study, and we know that it contains more than 100 trillion bugs of over 1,000 different species. That's why many individuals refer to us as a walking mass of bugs. We have 10 times more microbiome organisms in us than human cells. The gut microbiome is the driving force in immunity. The largest microbiome interacts mostly with the human host. It is the one that shapes the immune system in many organ systems, and the immune system of the host, of us as human beings, is shaped by the gut microbiome. Remember, the microbiome is functional. It's plastic. It changes with the environment. It changes with your diet and where you live. It's dependent less on health and disease, but does change over time. The microbiome also produces, many of the species of the microbiome produce the substrates or foodstuffs for the rest of this microbial organism content. And one of the other things that's importantly produced are short-chain fatty acids. They serve as an energy source, but they serve as the anti-inflammatory agents for local and systemic immunity in organisms, human organisms. So, of course, over eons of time, we hooked up with the microbiome species around us. We internalized them, we outsourced to them human immunity, we gave them a responsibility for generating anti-inflammatory immunity in the human host to protect us against the chronic diseases that have now become part of our lives. And they become part of our lives because over the period of time, we as human beings, as we normally do, we changed our minds about the relationship we wanted to have with this very important substrate within us, the microbiome. We decided we could do it differently, we could do it alone. And so we changed our minds about the way in which we lived with the microbiome. So the ways in which we've actually done that, the things that we've done as human beings to change the relationship with the microbiome and to allow our microbiome to become dysbiosed or different, is based largely on the environment in which we now live. We've moved from farm dwelling to city dwelling, and I can tell you most of the chronic inflammatory disorders, certainly asthma and allergy, are almost unknown in farming communities, certainly in children that live in close relationship with livestock. These diseases are unknown because there's protection from a normal microbiome in that way. But the three big offenders that have come into modern living that have allowed the microbiome to become dysbiosed, especially in the gut, are birth by elective cesarean section, antibiotic abuse in early life or unnecessary antibiotic use in early life, and not being breastfed. This cartoon reflects a dysbiosed epithelium with an abnormal set of bacteria on it. So once you now lose the content of a normal microbiome, whether it be the number of species being lost or a change in the proportion of the species that are reflected, you get this abnormality on the epithelium. The epithelium becomes disrupted. It becomes replaced, the microbiome becomes replaced by pathogens. The pathogens interact with a different set of pattern recognition receptors, and a whole pro-inflammatory response of cytokines and cells emerges. So I mentioned that one of the most important etiological problems for dysbiosis of the microbiome is elective cesarean section. And let me tell you what I mean by elective cesarean section. I'm not talking about the delivery of a baby. You go into labor, you go into hospital, and you can't progress, or there's CPD and baby needs to be cut out in order to survive. That's cesarean section, emergency cesarean section, which can't be helped and is life-saving. Elective cesarean section is what's happening in the modern world. It's where you decide to have your baby on daddy's birthday or granddaddy's birthday. You decide on a date for delivery of your baby. Often those babies are also born prematurely and have other problems. But what you don't get with elective cesarean section then is mommy's microbiome. You don't get exposure to the microbiome that is contained in mommy. And that's the problem, that there's no transfer of a microbiome to a baby through elective cesarean section. And so already at the time of birth, we start to set up a process of dysbiosis by elective cesarean section. So this illustration is an example of a number of studies which have now been published from around the world, from centers which collect a lot of data. Most of these are Scandinavian studies where everything that happens to you in life is noted and collected. And what they are able to show is there's a close correlation in all of these studies. There's a close association, and I use the word association because it's not etiology. We don't yet have a randomized trial to prove these associations. But there are many association studies that are showing that elective cesarean section is responsible for chronic inflammatory disease. If you are born by elective cesarean section, you have a greater chance of inflammatory diseases like asthma and allergy, but also other chronic inflammatory diseases. And in this study, what is reflected is that immune deficiency, which you would have thought was entirely a genetic disease, is increased by 50%. There's a 50% greater chance of having a PID if you are born by elective cesarean section. So the microbiome and the gut-brain axis microbiome is responsible for a number of factors when it goes wrong. For stress, both acute and chronic, for anxiety, loneliness, depression, even odor and our attraction to one another comes from the gut-brain microbiome axis. But it's also important in a number of neurodegenerative diseases. And I'm going to give you examples such as Alzheimer's disease, some examples from Parkinson's disease, and quite a bit about schizophrenia, because there's a clear example of the gut-brain axis going awry. So it's well established that there is now a gut-brain axis where the microbiome in your gut it protects against neurodegenerative conditions, and if you have a dysbiosed gut microbiome, you're at greater risk of neurodegenerative disease. Remember that the microbiome is not just bacteria, but it also includes viruses, candida, fungi, and parasites, and I'm not going to talk much to those species. This gut-brain axis is a very complex process. I've tried to simplify the idea that the microbiome becomes dysbiosed, pathogens start to release neurotransmitters that are dysfunctional, they enter the vagus and the HPA axis, and eventually cause neuroinflammation. But the process is much more complex than that. And so dysbiosis of the gut microbiome results in a whole process. A decrease in the short-chain fatty acids that are responsible for anti-inflammation, a decrease in the cytokine and cellular response that makes up anti-inflammatory activity. Autoimmunity also has a strong link to the gut microbiome. Clearly, if the gut microbiome is important in immune functioning, autoimmunity is going to be a phenomenon that we see in the dysbiosed microbiome. Disorders like type 1 diabetes, significantly associated with particular disorders of the or dysbiotic states of the microbiome in the gut. A recent study in children demonstrated that decreased or abundance of lactate and butyrate, those short-chain fatty acids produced by bacteria, are able to link with B-cell autoimmunity, and this is probably one of the reasons for the association with diabetes in a dysbiosed microbiome. Vitamin D is a vitamin that is widely spoken about at the moment, especially in the context of COVID-19, and everybody is on vitamin D. But many diseases are associated with vitamin D insufficiency. And it's quite clear that many of the dysbiosed microbiome bacteria in the gut are associated with vitamin D insufficiency. Particularly proteobacteria in the upper gastrointestinal tract, with significant reduction in Pseudomonas and E. coli species associated with an increase in bacterial richness. So there's this bidirectional relationship between the microbiome and vitamin D. Vitamin D is clearly important in immune regulation, demonstrating this complex association between immune regulation, the microbiome, and dysbiosis. Finally, in relation to the microbiome, we've always thought that our consciousness is rooted in biology, and we've always believed that it's our biology, our brains, in which we are conscious. But as you can see, consciousness also resides in the bugs, the microbiome that live in and on us. They also help to generate consciousness in the human being. A major reason why we need to start to think about protecting our microbiome. The real question now is, what can we do if we've lost part of our microbiome for the various reasons I've said? Can we do something artificially to restore the microbiome? Now, there are a number of studies that have shown that there is a probiotic effect in helping to reestablish certain components of the microbiome. So, for example, if probiotics have been shown to stabilize the epithelium and create at least an environment in the gut and the skin and the respiratory tract where there is no more leakiness to pathogens. So at least probiotics have been able to demonstrate that. Multiple studies have shown now that there's a clear role for probiotics and prebiotics in the prevention of infections in children attending daycare centers, for example. A significant number of studies have emerged to demonstrate that probiotics are useful in preventing upper and lower respiratory tract infections in these individuals, as well as gastrointestinal tract infections. And at least 12 studies now have identified that probiotic supplementation to children in daycare centers is able to prevent a large number of gastrointestinal and respiratory tract infections in this population. So the value of probiotic supplementation in respiratory tract and gastrointestinal infections, I believe, is now widely established. There are a number of studies, this meta-analysis, for example, reflecting that probiotic use in a number of studies able to reduce the number of respiratory tract infections in children, and a double bang for your buck, reducing the number of antibiotics you need as a young child if you have an artificial probiotic at the time of a respiratory tract infection. So not only do you not have the infection, or the infections are reduced, you also don't need an antibiotic, which destroys the microbiome. Probiotics are clearly effective in controlling various forms of diarrhea, including antibiotic-associated diarrhea, traveler's diarrhea, and probably diarrheal diseases in children caused by rotavirus. In fact, what's not well published and what not well recognized is in fact that the biggest bang for your buck with the probiotics is in the premature infant. A number of studies now generating a meta-analysis that shows that there's a significant survival advantage for the preterm infant given probiotics. This is probably an effect on necrotizing enterocolitis, but certainly this is not well known in the pediatric community. All right, so we've spoken a lot about the microbiome and probiotics. I just wanted to remind you that there are also other important constituents that come from human breast milk, now are artificially replaced in formula feeds that play a significant role in generating anti-inflammatory events in the human baby. And one of these is the human milk oligosaccharides. They play as important a role as the probiotics in terms of creating normal immunity for young children. So in summary, ladies and gentlemen, what I've mentioned to you is the association between the gut, the microbiome in the gut, and the brain. I've hopefully been able to share with you during this presentation the important relationship between what happens in the gut in relation to the microbiome and the brain. That the gut microbiome is responsible for many of the things that we take for granted, like human emotion, human connectedness, but also responsible for a large number of neurodegenerative diseases when the gut microbiome becomes dysbiosed. And many of these neurodegenerative diseases are real, and many studies are now starting to look at the possibility of using artificial probiotic replacement to impact on these diseases. Those are not clearly evident yet. Those are not studies we can hang our hats on yet. So I'm not suggesting that we start to replace probiotics in individuals who are at risk or have one of these diseases. But I do believe that in time we will have a pill containing a microbiome or a probiotic that will take care of many of these chronic neurodegenerative diseases. So lastly, let me thank you for your attention during this presentation. And I just wanted to thank my team who work with me and my mentors. Thank you very much for your attention. Goodbye.